Welcome. Today, we're joined by Francesca Barone, who is the CSO of Candel Therapeutics. Ticker on the NASDAQ is CADL. Francesca, thank you so much for joining me.
Oh, thank you for having us, actually.
Absolutely. Before we dive into the nitty gritty and look under the hood, give me a quick brief overview of what is it that your company does.
So, Candel is a company that is based on the construction of viral immunotherapy. These are therapies that leverage the capability of the viruses to infect tumor cells, to kill tumor cells using different mechanisms, release tumor antigens in the microenvironment, and elicit an immune response against the tumor itself. So we are actually a clinical stage company. We have two clinical candidates, CAN2409 and CAN3110. Those are two very different type of viruses, both of which have got multimodal capabilities of attacking tumor cells. CAN2409 is in clinical stage development, is in phase 3 in prostate cancer, in phase 2 in pancreatic cancer and in lung cancer, and then CAN3110 instead is in phase 1 development for recurrent high-grade glioma.
Now, I recently read about your enLIGHTEN discovery platform. Can you tell me exactly what that is? And also if AI is involved with that?
Yes, it is. Actually, this is the newborn research endeavor within Candel. We're very proud of it. It's a completely new type of platform for the construction of these multimodal viral immunotherapies. It's made of two components. We leverage the capability of the viruses to have tunable features. And so we use these features, like, for example, the capability of virus to replicate, to infect cells, to induce oncolysis, or killing of the tumor cells, to release antigens, and to inflame the microenvironment of the tumor. And together with that, we also leverage the capability of the viruses to deliver payloads, so proteins, antibodies, to the tumor microenvironment. The different things about enLIGHTEN is that we do use AI for two reasons. So, computational biology and machine learning are used both to pair the virus with the right payload, so with the proteins that we deliver, both and to select the proteins that we put inside the virus. The virus backbone that we use is HSV, so herpes viruses, and they've got a very large encoding capability, especially when we modify the virus backbone to encode different proteins. And we rely on the fact that the tumor microenvironment is very diverse, so different condition or pairing with different type of therapeutics might require a different combination of proteins to be encoded in the virus. And we don't want to do that just by chance, but we use AI to select the right proteins to put inside the virus.
Now, it's interesting that you have AI in a platform in terms of therapeutics. I love seeing how it's ingrained in everything that you're doing. You also recently had a partnership with UPenn. What does that partnership look like? Love to know the scope of it, and also how enLIGHTEN is going to boost the efficacy of CAR T therapies.
Yeah, so this was actually the partnership with UPenn was unveiled at the same time in which we unveiled enLIGHTEN. And I think you really nailed it when you say like how we can boost the efficacy of CAR T cells. So CAR T cells have been, and UPenn has been a leader in CAR T cell development with Carl June. They've been fantastic in treatment of blood tumors, like leukemia, lymphoma, but they've not been as efficient in treatment of solid tumors. And the reason resides in the fact that the CAR T cells very often, when they reach the tumor microenvironment, if they can reach the tumor microenvironment, become exhausted and they are lacking survival factors and mechanisms that really keep them in the tumor microenvironment to be able to be efficacious against the tumor. So what we're doing here is that we're designing a virus, a specific tailored virus, to be paired and co-delivered with the CAR T cells that will enable the tumor microenvironment to be more amenable to the efficacy of the CAR T cells. So the virus will do several things at the same time. It will release the antigens, so they are able there to be exposed for the CAR T cell to recognize them. At the same time, what it will do is that it will favor the recruitment of the CAR T cells. It will favor the survival of the CAR T cells. Will destroy things that are present in the tumor microenvironment. They will suppress the activity of the CAR T cell. So it's really a design therapeutic to be co-delivered, we delivered actually in the tumor microenvironment itself to favor this activity and to boost the activity of the CAR T.
Now, how many active candidates are currently under development in that platform?
So the platform, we have unveiled two candidates already, two experimental candidates, we call it like that. One has been, the data have been released at SITC, and this is a virus that actually encodes a single protein. This is a protein that interferes with the crosstalk between SIRF1 alpha and CD47. The SIRF1 alpha CD47 pathway is a pathway that is involved in the anti-tumoral response against the immune system. So it's very well known, and many people have tried to target this pathway with systemic delivery of drug that will interfere with this crosstalk. The problem is that there is a lot of systemic toxicity from this type of drug. So what we've thought is that if we can deliver this antibody, in our case, is something that interferes with this crosstalk in the tumor microenvironment itself, we can reach a very high concentration locally without having the problem of the systemic activation of the systemic side effects related to the systemic exposure of the drug. The other advantage is that obviously we still leverage the ability of the virus to kill the cell and expose the antigen. So again, it's a multimodal therapeutic. This was unveiled at SITC. And then we're very excited about the second investigational therapy that we are developing. And this is something completely different. This is a virus that will be able to induce tertiary lymphoid structures. So tertiary lymphoid structures are like little lymph nodes, aggregates of lymphocyte, extremely organized aggregates of lymphocyte that form inside the tumors. The presence of these structures has been associated with better prognosis for the patient in which they were detected inside the tumors, but also is associated with better response to checkpoint inhibitors, so other systemic immunotherapy. So everybody wants to form tertiary lymphoid structures. And I actually carry a lot of experience from my previous life in autoimmunity in my academic career, where I was trying to destroy these structures because actually in autoimmunity, they're very deleterious. And so flipping the coin between autoimmunity and actually immuno-oncology, we are actually leveraging this knowledge, the knowledge of the factors that are involved in the formation of the TLS, to create this single therapeutic that will enable the formation of these tertiary lymphoid structures. And this is very unique because, as I mentioned to you before, you leverage the ability of the virus to release antigens, to inflame the microenvironment, and then to deliver genes. These are all things that you need to form a TLS. So you need to have a lot of antigen present and exposed within the context of a very inflamed tumor microenvironment. But you also need the genes that are necessary for the formation of these structures. And so we have proved that we can do that in animal models, and we'll be very excited to test this in future clinical development.
Well, there's so much happening with this new endeavor of yours. I'm curious, what is the most promising area at the limits of current cancer research, in your opinion, of course?
I think that the formation of TLS is something that people really look at because it's something that the academic community has been studying and looking at for quite a while. And I think if we're going to be able to demonstrate that this has got an antitumoral effect in humans, it will be extremely important for tumors that are resistant to checkpoint inhibitors, classical tumors that do not respond to immunotherapy, like sarcoma, for example, extremely cold tumors. The future, I think, a really different tier of cancer therapy is the combination therapy of checkpoints or other immunotherapies together. And I think that what we bring with enLIGHTEN is really a rational, data-driven approach to the selection of the pair therapeutics that can be delivered and overcome mechanism of resistance, both for immunotherapy or cellular immunotherapy, like, for example, CAR T cell and K CAR cell and so on. So we are very excited about this.
We've talked so much about what, partnerships that you've started with, what you're currently doing. I'd love to get your final thoughts on what we can expect next from the enLIGHTEN discovery platform.
So I think that from the clinical development, we will hope to be able to bring these experimental candidates to patients. But from the discovery point of view, this platform is really open for collaboration. So what we can deliver is really, as I mentioned before, a rational approach to selection of potential candidates that will overcome problems with current therapies. What we want to do also from our own discovery point of view, we want to explore areas that are very difficult to achieve with systemic therapy, like, for example, metabolic modification of the tumor microenvironment. Those are very difficult to, the type of therapeutics that are very difficult to administer systemically for the potential of target effects. And I will be very excited about the possibility of modifying the metabolism of tumors with very selected targeted approach like the one we have, again, as a monotherapy or in combination with other therapies.
All right. Well, I look forward to hearing more about it. And of course, seeing how your partnership with UPenn pays off in the short and long term. Thank you so much for your time. I appreciate it.
Thank you for having us.
Absolutely. That is Francesca Barone, who is the CSO of Candel Therapeutics. Ticker on the NASDAQ is CADL.